Okay, thank you very much for the invitation. I think this has been an amazing meeting so far, going from ligands to proteins. And I think it's just great how we can see AI apply to all these topics. Today, I will just talk a little bit of ligand binding, or how we can predict conformation of ligands using deep learning. I think it would be interesting because, as you will see in the talk, I took many concepts or ideas that have been applied for protein conformation or protein folding prediction and apply it to ligand binding. And I think this is something we can keep doing, like just exchange ideas between fields and keep growing together. This work was also a collaboration with, of course, Massen and Jörg from Janssen and Antonio from Imperial College at the engineering department. The way I want to start the talk, it's thinking more or less a little bit how a computational chemist thinks when he has like a new ligand that is bound, or he predicted the conformation to a protein, the ligand conformation into the protein. And usually when they saw something that is strange, they start thinking, is this real? Is this just an artifact of the method? And one way they usually do it, for example, we have this conformation, and they want to see if these two interactions here with the pink lines are real or not. So usually what they do is they just go to the basics. So they just take this fragment, take the amino acid or the part of the binding site, and they just go to the PDB and see how many times this interaction it's there. So how frequent is this interaction? In this example, you can see that this interaction at 2.5 angstrom seems to be quite, this is just an example, it's not real data, but in this example, it seems to be quite frequent, right? You have many times that this interaction is repeated in PDB, so then you can think this is something that it's real. The other interaction, for example, you do the same, go to the PDB, but here the distribution, it's a bit different. So you can see, yeah, it's something you can see at 2.5 angstroms, but it's more probable to find this interaction at 4 angstroms. And this is something that we usually do. So I wanted to take this idea, this way of thinking, and translate that to deep learning and how we can use this data to predict, or these ideas to predict the ligand conformation. So here, it's impossible to do this analysis for all possible ligands and all possible binding sites, because it would be just huge and enormous amount of data. So taking all fragments and all amino acids is just impossible. But one way we can do it's actually using DeepDock or machine learning, deep learning, to really learn these patterns. So we can learn how each fragment looks like and how each part of the binding site looks like, and then predict the distance probability distribution for each pair. And using these distributions, then we can score and see if these interactions are possible, not possible, or if it's something we'll expect or not. So for doing this, the first real challenge actually was to find a way how to describe the binding site and, of course, the ligand. I think at this moment, the biggest challenge is how to describe the binding site. But luckily, a couple of years ago, last year, actually, 2020, this group just, they published a way to describe protein surfaces using a mesh. So you can see that they calculate the molecular surface of the protein, and then using this molecular surface, they just describe this surface into a mesh. So this is, I think we can see, everyone has seen this in any software. It's possible to see this. But the thing they did is actually in each node of this mesh, they calculated certain properties. So they encode properties related to electrostatics, to hydropathy, hydrogen donor and acceptor, and a shape index, which is how the curvature looks like at this specific point. They they did this. It's available. You can download it from GitHub. We did some modification just to make it run for our purpose. And this is the way we use to describe the ligand, the protein surface. Now for the ligand, as you know, there are many ways to describe ligands, from fingerprints to now graphs. We choose graphs just because I think for my mind, it just really captures the topology in a better way. And a way to describe a ligand into a graph is just you take the ligand, the molecular structure, and each atom then will be a node in the graph, and each bond will be an edge connecting two nodes. Then you have, in this case, you have two matrices. So one matrix describing each node and which type of atom is there. For example, here you see node number 1 is an oxygen, node number 2 is a phosphorus, etc. And this is a binary notation. So where you see colors, it's 1, and the empty spaces is just zeros. And similar for bonds. So you have each edge, so bonds connecting node number 1 to node number 2. And then you see if it's single, double, triple, or aromatic. Again, this is ones and zeros, and this is the input for our model. So the model, actually, it's divided in two phases. The first phase, it's what we call feature extraction. So we gave the information about the ligand in these two matrices, as I mentioned before, and about the target, which is also two matrices, one about nodes with all the properties I mentioned before, and the other one with information about the connecting edges. So here, the connecting edges, the description is just the distance between each edge in each coordinate, just to have a little bit of information about the 3D information of the 3D position of the mesh in the space. This is fed to two individual graph convolution networks. It starts with a normal graph CNN, but then after 3, we did residual blocks just to be able to do more convolutions if they are needed without losing the gradient. So in the end, what we will have, it's a representation of the node, but now this representation is not only about one atom, but about one atom environment, and the same for the mesh. So I want to explain a little bit more in detail this convolution part, because it might be a bit tricky. So if you remember the last slide, we can represent atoms as a 1 and 0 vector. So in this case, let's take atom number 4, and this will be just a vector of zeros with a 1 when it indicates it's a carbon. So at first convolution, what we will do is take the molecule and share information about the neighboring nodes and the connecting edges. So after this step, the vector that will describe atom number 4 will not be only a binary vector, but now will be a continuous vector, which has information of all these 3 atoms, so the neighboring atoms. If we do this again, okay, now we share information about other neighbors, so the neighbor of the neighbors, and the resulting vector will have more information about a bigger part of the molecule. And we can do it again and again. So if we do it a third time, we have information about larger portion of the molecule. In this case, this specific molecule will be completely covered with 3 convolutions, but the bigger the molecule, they might be different. So this step, it's done for all atoms at the same time. So basically what we will see is just you give information of single atoms. At first convolution, you have the first round of fragments, second convolution, a bigger fragment, and third convolution, a larger fragment. So for those cheminformaticians here joining today, this might be quite similar to what we were used to use before with ECFP fingerprints. So here you can think this is an atom environment, which is centered in atom 1. This will be the atom environment centered in atom 2, and so on. So the idea is quite similar, but here each fragment, each atom environment will be described by a continuous vector. So now the same idea goes for the mesh. You just start with one point in the mesh, and then you start giving information about the larger portion of surrounding nodes in the mesh. So this is the idea behind. Once having this feature extraction, so we can talk about here about atom environments, and here about a portion of the surface in the binding site. What we do, it's a pairwise concatenation. So basically every atom environment, which every point in the binding site. And with this, and finally, we use this as the input to the mixture density network. The mixture density network, what it's doing is actually predicting a set of parameters that we will use to describe a distribution. So this distribution will be a mix of Gaussians, and the thing that will be predicted by the model will be a set of means, a set of standard deviation, and the factor, the coefficient used to mix these different Gaussians. We use 10 Gaussians in this case. So the idea is that you will have a distribution, a mix of Gaussian for each pair of ligand target nodes. So for example, in this example that I will be talking about this talk, it's I think we have around 350 nodes for the target and 9 atoms or 9 nodes for the ligands. So in total, we will have something around 3,000 pairs. And because of that, we will have 3,000 distributions, each distribution having the information of each ligand node with each target node pair. So once we did this, actually something that I want to mention as well is that the input for the ligand here, it's just 2D. This is important. But the information that we input here is 3D. So we are encoding the 3D information feed here in the edges. And what we want to predict is the distance in a 3D conformation between each part of the ligand and each part of the target. So it's not a completely 2D model, but it's not a 3D model either. So how can we just jump from distributions or likelihoods to a scoring function? So the way we do it, it's basically we take the 2D information of the ligand, the 3D information of the target, put it in the model. The model will predict a lot of distributions for each pair of nodes. And then we can score a 3D conformation. So we just take the 3D conformation, measure the distance between, for example, here, these atoms that I have here, and then go to the respective distribution for this pair of atoms and see how likely it's to find these two atoms next to each other at that specific distance in the whole training set. So in this case, you can see some pairs are more frequent than others. We measure the likelihood, and of course, this is done across all the possible pairs. So in this case, the 3,000 pairs, and we just sum it at the end. So we just add all these likelihoods together to have a final score. So this is the idea behind the model and how it's working. And now I want to talk a little bit of how we wanted to validate this model and see if it's really working or not. So for that, we took the CAS 2016 data from this challenge. So this data has 258 diverse complexes, and it's divided in 4 tasks. So the first 2 tasks are scoring and ranking, but in this case, we are not using it because we are not doing activity prediction. And then we have docking and screening. So the docking task actually, this challenge or this data comes with 100 decoy conformations for each of the 258 ligands. And the goal of the task is for the scoring function to find the conformation that is closer to the real. So we score the 100 decoys and choose the best one, the highest rank, and this should be quite similar to the real conformation. Then we have the screening task, which is actually divided in 2. So the forward screening task, which is you have the target, And then you have a set of 28,500 conformation. So these conformations are coming from 100 conformation of each of the 258 ligands. And the idea is to find the true binders for this specific target across all these conformations. And then you have the reverse screening task, which is for each ligand, try to find the correct target. So it's a reverse docking kind of. And for this, for each ligand, you have 57 possible protein targets. So these are the results. Of course, we just did it for DeepDock, and then we use the data that has been published before for many other scoring functions. Some of them are very, very common, like autodock or glide. I think you have also leak score. So there are many scoring functions to compare here. You can see that DeepDock here did like in the top 5 for the docking power, but I will say this is really you know, they are very close, all of them. For forward screening power, DeepDock did really, really well. So scoring the first compared to all the other scoring functions. And for the reverse screening, also our method did quite well, so finding the right targets for the right ligand. So we were quite surprised and excited for this. And the idea was like, okay, we have a kind of a scoring function coming from deep learning approaches. If we can score a conformation, that means we can use it to create a conformation, right? So we just took the idea from docking. I would say you have the rotatable bonds for each ligand. Sorry, you will describe each conformation based on the rotatable bonds of the ligand. You have 3 possible parameters of the translation of the ligand in the Euclidean space, and then you have the Euler angles of the ligand. So basically, if you take the ligand as a fixed body, it's how you rotate this body in space. So with these parameters, you can actually describe the conformation of all the conformations for that specific ligand. So we use this, we score this specific conformation using our approach, the DeepDock, using the likelihood potential, and then we use this to optimization algorithm. So in our case, we use differential evolution, but that doesn't mean it needs to be this this algorithm all the time. Actually, if you work yesterday, you can see we can apply also the Firefly approach. We can use a particle swarm optimization, so you can choose your favorite here, and then go back to a new conformation, score it again, and just continue this optimization process until it converges. So this actually looks like, this process looks like this. So you start in a random place in your protein or in your mesh. As you can see here, the likelihoods are quite low for this conformation. And you will see here how the score increases. So we start getting very fast to a position where the ligand it's in the actual binding pose. So I think I will just stop it there. So very close to the end, it's almost converging. You can see that the distances gets very quickly to the highest, to the point of the highest probability, most of them. Here I just took 3, just for example, but this is done across, again, across the 3,000 pairs for this example. So having this, okay, so if we can score, we can optimize the conformation, then how well are we doing for this optimization, for this conformation prediction? So what we did, it's we redock basically, or create the conformations from all the 285 compounds in the CASP dataset. As you can see, most of them converge quite well. So when I say successful, it's the optimization finished successfully, and unsuccessful means the optimization didn't finish with the parameters we use. So most of them, so 225 out of the 285 finished completely, and with a mean RMSD of all of 1.87, which is I would say, quite acceptable. Something interesting is that when we analyze why compounds are not converging or finishing the optimization, it's because the ones that are very large with many degree of freedom, it's very difficult for this particular optimization algorithm to really optimize them. So we need more time or more iterations or simply another algorithm. Similar results we did with a test set, which was not included in the training set, of course. This is much larger, so it's 1,367 compounds, for which around 900 finished successfully. Results are quite similar, I would say. And here we also notice that above 10 rotatable bonds, so the degree of freedom for the optimization is too large for this algorithm. So maybe trying new algorithms which can deal better with this, it's a good option. And also something to keep in mind is that these optimizations are not as fast as a docking software, because of course it's not optimized for that. Hopefully we can get it to run faster, but at the moment it takes few minutes or half a second, depending on how large is the molecule. So as you see during this talk, we have many ideas from AlphaFold, for example, to using this distribution, learning this potential. So just something that I want to take home is that it's possible to learn potentials from ligand and target. So not two bodies now, not only one. Do this with 3D information. The potential that we learn actually perform very similar or better in some cases than very established scoring functions like glide, autolog, etc. So might be a good alternative. You can couple this scoring function with optimization process. But I think the key message I want to give today is this is just the start of how AI can be applied for predicting ligand binding conformation. Hopefully, at some point, we can just do it directly, make a docking or virtual screening super fast. And I think this is possible and it's coming in the next years. So with this, if you want more details, just go to the publication here, it's a preprint, or just drop me a tweet here. I'm happy to respond. And thank you very much for your attention.